This next class of chemotherapeutic drugs are referred to as the anti-tumor antibiotics. So these are chemicals, compounds harvested from nature, made by bacteria that are actually so potent that they actually can kill mammalian cancer cells. And the first one we're going to talk about here is doxorubicin, which was isolated from this very interesting castle in Italy from a bacteria called Streptococcus paucides. This novel compound was first named daunorubicin for the ancient pre-Roman tribe that inhabited that portion of the Italian peninsula. The trade name was adriamycin, after the Adriatic Red Sea. And so a lot of these compounds in this entire lecture series actually come from nature. So it's an interesting kind of side story. So these anti-tumor antibiotics can kind of be subdivided based on their chemical structures. The one we're going to talk about are the anthracycline antibiotics, which include daunorubicin as well as doxorubicin. Anthracyclines, I always remember anthracyclines, specifically with doxorubicin, as a DNA intercalator, basically between the unwinding pairs of the DNA double helix molecule. And these drugs are specific to topoisomerase type 2. And the way I remember it is doxorubicin has two O's, and so topoisomerase also has two O's, and then the number Roman numeral 2 fits in there. So that's our physical target. We're going to prevent the relegation of DNA during DNA replication. We're going to induce double-strand breaks, which really kind of put a lot of stress on the DNA molecule itself. It's torsional strain, as shown here, or as written there. You can see the generation of oxygen-based free radicals. And that actually contributes to single-strand breaks. And so this is actually quite a potent drug, because now you can, through kind of two different mechanisms of action, you can produce both double-strand and single-strand breaks. This basically causes the DNA molecule to be quite unstable, and during the unwinding process, you actually have a prevention of relegation or joining back of the double helix copies, and that will lead to further breaks. We use anthracyclines such as doxorubicin in a variety of tumors, both solid and hematologic in origin. In terms of the toxicity, we'll see myelosuppression, which is usually dose limiting. We also associate these drugs with very potent cardiotoxicity. They are vesicants, so you have to be careful about where you're going to inject them or administer them. They can't be put in every compartment. You can see kind of a delayed, both acute and delayed, nausea and vomiting, which is often dose dependent. And the one thing that the STEP does like to talk about in terms of side effects or interesting aspects of these drugs is that they're associated with a discoloration of body fluids. So doxorubicin is associated with a reddish-orange discolorated body fluid. Metaxatrone is another drug that's associated with a blue-green color change, and so it's kind of a high-step yield memory device there. The cardiotoxicity is the main highlight for doxorubicin and other associated anthracycline antibiotics. And the mechanism of cardiotoxicity we think comes from the oxidative stress that this compound produces in terms of its ability to produce superoxide and free radicals. The heart's especially sensitive because it has low levels of antioxidant enzymes there anyway. And those excess free radicals can accumulate within the blood vasculature and kind of get deposited within the myocardiocytes and cause dramatic myocardiocyte cell death via apoptosis. In terms of the cardiotoxicity, there's an acute reaction that presents within about a week of that first anthracycline dose, and you can see that as evidenced by EKG changes or even pericarditis. And so you can see arrhythmias generally reverse within a few hours. There can be some ST wave abnormalities, and that will sometimes reverse back to normal within 1 to 2 weeks after that first dose. So that's kind of the acute cardiotoxicity profile. The chronic toxicity profile is a little bit different. We can see dilated cardiomyopathies with the production of congestive heart failure. This may be with a reduction in ejection fraction, as shown below. It can be less than a year or after a year from the last dose of the anthracycline antibiotic. The ejection fraction might decrease by 20 to 50%. And then, of course, the patients would have symptoms of congestive heart failure. So you can actually somewhat prevent some of these anthracycline-induced cardiomyopathies. Of course, you must give all patients basically some baseline cardio imaging to make sure that the heart is relatively strong and capable to withstand some of these drugs. You can also co-administer something called dexamethasone, which is indicated for the reduction of, specifically for the reduction of doxorubicin-induced cardiomyopathies. This drug basically is a chelator of some of these more potently reactive divalent ions and ferric acid moieties, which basically kind of prevent the free radical formation. In order to prevent the cardiotoxicity, you typically give 10 times the doxorubicin dose. And of course, there's other dosing guidelines there as well to prevent extravasation. Okay. Other drugs that are in that category have less cardiotoxicity, which is why they're clinically relevant. But doxorubicin is still, as horrible as it is in terms of its side effect profile, it's still kind of a really important chemotherapeutic drug because it's in so many different regimens that are out there.